Welcome you all back to SBT 114, Carbohydrate and Protein Biochemistry. We started last week with proteins and particularly looking at the building blocks of proteins, which are amino acids. So we took a look at the structure of amino acids and looked at some properties of the amino acids. Today we'll continue to start putting the amino acids together to form proteins. And in about maybe 1 hour, 30 minutes, we should be done. And the floor will be open for you to ask any questions that you would like to ask. Or if you have any contributions, you can make those contributions. Just like we did the last time, the charts is available. So if there is anything, you can quickly type in the chat. I'm monitoring it. I'll see it and I'll respond. So even throughout the lecture, if you have anything, you can use the chat and I'll respond to you. Alternatively, you can note down any concerns, and at the end of the lecture, when I open the floor, you can put up your hand and I will call your name, enable your microphone, and you speak, then I respond. As you can see, I was telling you the last time that this semester is very short. We're having 10 weeks of lectures. This is the second week. And you see, we are continuing on and on. Hopefully, after 5 weeks, we should be done with the protein part of the course. Because as you can see, the title says Carbohydrate and Protein Biochemistry. And we're starting with the protein biochemistry. So the plan is we divide it into two so that we use 5 weeks for proteins and use 5 weeks for carbohydrates, roughly that way. And I told you the last time, don't wait till the last minute before you want to cramp everything into your head to write exams. So the things we did the last time, you must have revised them. And then the ones we'll do today, you revise them. As time goes on, I will give you some self-assessment questions as well, so that you can be trying your hands solving those questions, preparing yourself for the exams throughout the semester before the examination week comes. So don't wait. You must do biochemistry every day, little by little, and everything will be fine. Right. So we dive into the formation of the proteins, and this is about the formation of peptide bonds. This is something which you have talked about before in primary school, in junior high school, senior high school. You talk about peptide bonds. Sometimes you refer to it as amide linkage or even amide bonds. These are the bonds or the linkages between the amino acids that you find in the proteins. And you see here, what is being depicted is that you have an amino acid. So on the left-hand side, with a carboxylic acid group, what we refer to as the C-terminus. The carboxylic acid end is a C-terminus, and the amine end is what we refer to as the N-terminus. Now, what you see is that you remember the last time we saw that it is the side chain of the amino acids that distinguish between the various amino acids. So that the carboxylic acid end is the same, the amine group is the same, but the side chain, which is being represented by this R1 for the amino acid on the left-hand side, and then the second amino acid, you have R2 as a side chain. So these are two different amino acids which are adjacent to each other, such that you have the carboxylic acid group of one of the amino acids and the amine group of the other amino acid in close proximity, that there can be a reaction between these two amino acids. And that reaction is what will result into the formation of the peptide bond. So the peptide bond is what is shaded in the yellow. You see the carboxylic acid end that is having a bond between the carbon and the nitrogen there. That is the peptide bond. And that peptide bond is formed between the amine group of one of the amino acids and the carboxylic acid group of the other amino acids, That's why you have it, all being shaded yellow for you to see the group which is involved in the formation of the peptide bond. Now, what kind of reaction is this? What kind of reaction is this? Now you can mistaken to think that the reaction between the carboxylic acid and the amine group is an acid-base reaction, but this is not an acid-base reaction. So somebody is saying that he can't hear. Emmanuel, I think maybe you are the only one who can't hear. If you can't hear, the problem must be from you, because right from the beginning, I confirmed that everybody could hear me and see the screen. So if you can't hear, you might want to check the volume of your device. And also, if not a volume, maybe you should use a speaker. Sometimes some of you just leave the device on its own audio without. So you change it to speaker and you can hear well. All right. So Ahmed Elias, I said you have a carboxylic acid group, that is the COOH, and then the amine group, that's the NH2 or NH3+, that's the amine group. And you have a reaction between the carboxylic acid group of one of the amino acid and the amine group of the other amino acid. Now, I was saying that you can mistake to think that this reaction between the carboxylic acid group and the amine group is an acid-base reaction, but it is not. This reaction is not an acid-base reaction. Why is it not an acid-base reaction? It is not an acid-base reaction because if it were an acid-base reaction, you have a carboxylic acid group that would have been giving out hydrogen ions. And the amine group should have been accepting the hydrogen ions. But this is not what is happening in this case. In this case, what you see is that a hydroxyl group is rather coming from the carboxylic acid group, binding with one hydrogen from the amine group, forming water. So water is released in this process. What you can refer to as some form of dehydration, or even the reaction itself is referred to as a condensation reaction. Just like how we say, the meteorologists talk about, say, rain as condensation. So you have a formation of water coming from this reaction. Water is removed. That's why it's also being referred to as dehydration. So this is a condensation reaction, but not an acid-base reaction. You want to keep that in mind. Now, this reaction that is between two amino acids can continue to form. So if you have this first reaction, which is forming this peptide, you have a dipeptide. A dipeptide consists of two amino acids. So if you have other amino acids that are joining this chain, you can have a tripeptide. So that will be a peptide that consists of 3 amino acids. You can have tetrapeptide, pentapeptides, etc. And usually we refer to peptides with amino acids from 2 amino acids up to 20 amino acids. We group them as oligopeptides. And above that, we refer to them as polypeptides. So sometimes you can hear the term peptide, which is referring to proteins as well. A peptide is the same as a protein. So you have amino acids coming together to form peptides, which is also referred to as proteins. Right. Now, I think Paul Abuchi shouldn't be asking this question. What links dipeptides together? I mean, this is what we are referring to. These are peptide bonds which are forming between amino acids. So, I mean, what do you mean by what links dipeptides together? All right. We can continue and look at what we refer to as different levels. We have 4 different levels of protein structure. Now, what we were referring to just before, that is having amino acids, having peptide bonds between them. What it means is that since we know the amino acids which are present in proteins, you know, the last time we studied these amino acids, glycine, alanine, and all the others, phenylalanine, tyrosine, tryptophan, leucine, isoleucine, all those, which means that you can actually talk about the structure of a protein by referring to the sequence of amino acids which is present in the peptide chain. So, for instance, you have this primary structure which is being shown here, depicting that you have lysine, that's an amino acid, which is linked to another lysine, and that is linked to glycine, and that's also linked to another glycine, which is linked to leucine, and that is linked to valine, and which is linked to alanine, and that is linked to histidine. Now, when you talk about the structure of protein, a protein chain that way, referring to the sequence of the amino acids which are in a protein chain, what you are talking about is the primary structure of the protein, just referring to the sequence of amino acids in the protein chain. Then you have the second level of protein structure, which is referred to as the secondary structure. One of the structures you are seeing here, we'll go further into it, and you see it's not only an alpha helix, which you see. So you have from the primary structure, you have some ribbon-like structure which can form, which is referred to as an alpha helix. But this is just one of the secondary structures. When we go next, you will see that we can have other forms of secondary structure. And then we have a tertiary structure, which is the folding of a protein in space in say a 3-dimensional structure, such that that protein is having a particular form, a conformation, which will now be active to be acting as a particular biomolecule like an enzyme or a neurotransmitter, a hormone. So any of these proteins will be having some particular structure in such a way that it makes that protein to be active. Now, this kind of folding in a 3-dimensional structure gives you form such that you have multiple secondary structures that are identifiable in the structure of the protein. And that is what it's referred to as a tertiary structure. We'll look at it further. And then a quaternary structure will be having a protein that is having, say, more than one subunit. So you have proteins, let's say 2, 3, 4 different proteins that are coming together in a complex, acting as one. An example is the hemoglobin molecule, which is having 4 different subunits coming together, acting as one. That is what is referred to as a quaternary structure of proteins. Let's take a further look at these things. So let's take the secondary structure, because the primary structure is simple. It's well understood already that you just have amino acids that are having peptide bonds between them to form these peptides, dipeptide, tripeptide, tetrapeptide, and so on. Now we take a secondary structure. The first we're looking at is the alpha helix. I mean, the one we were looking at in the structure before, the alpha helix. We have two of them, so we'll take a look at the second one as well, how it looks like and what is involved in it. I don't know why some people are saying they cannot hear me. If you can't hear me, the problem is really, I don't think it is from me, because I think the rest of the people can hear me clearly. So if anything at all, the problem should be coming from you, so that you want to solve that problem quickly. All right. What is the alpha helix? So you have the protein structure, which is forming some ribbon-like structure in such a way that you have the carboxylic acid group of some amino acids and the amine group of other amino acids coming in close proximity, such that you have hydrogen bonds that are between that oxygen of the carboxylic acid group and a hydrogen of the amine group. So the dotted lines that you see, these are showing hydrogen bonds that can exist between carboxylic acid group of one amino acid and hydrogen of the amine group of another amino acid. And the structure is like a ribbon. You can see this structure in more detail on the left-hand side, where you have C terminus labeled there, which is referring to the carboxylic acid group end of the peptide, and the N terminus is the amine group end. Because of course, if you have a peptide which is having an amine group ending at one side, a carboxylic acid group will end at the other side. And the dotted lines, the hydrogen bonds, are being shown between oxygen and hydrogen of this structure. You can look at the key down there. These the black balls refer to the carbon, the red, oxygen, teal, nitrogen, violet, which is the R group, and then you have this gray ball, which is referring to hydrogen, just to see it much better. Nobody should be drawing such a this kind of thing is just to show some form of a molecular modeling, ball and stick model, to show this kind of hydrogen bond which can exist in the alpha helix structure of proteins. It's not a thing that anybody should say is drawing. If anything at all you want to show simply in a diagram the alpha helix structure, I mean, what is on the right hand side is just enough to show that. I mean, what is needed is for you to show that there's an interaction, a hydrogen bond interaction between oxygen of a carboxylic acid group and hydrogen of an amine group. That's what is needed. And so you have a. So I really don't know. Rose Foley-Sanu keeps saying she can't hear me, and she's the only one writing. So those of you who can hear me, maybe you would like to help her, so that whatever her problem is, you can help her solve it. So if there's any solution, we can write it in the chat for her, so she will do that, so she can hear me. All right. Now we go to secondary structure. Another secondary structure, what we refer to as beta sheets, the beta sheets. Now you see that in this case, even though the bond is is still a hydrogen bond, which is between oxygen of a carboxylic acid group and hydrogen. Even this hydrogen can either be the hydrogen on the alpha carbon or the hydrogen of an amine group. But in this case, the structure which is formed is not the alpha helix structure which we saw earlier, but this forms some zigzag structure such that you have the folding of the protein in an anti-parallel and a parallel form. So you see this time around, you see an N-terminus below here, and the protein goes up, folds, comes down, then you are going to have an N-terminus here as well. I mean, below here will be an N-terminus. That is, if the structure should have ended here, this will be an N-terminus. So that is a parallel structure. I mean, the first look that way is a parallel structure. And from the N-terminus, you have here going up again, C-terminus. So you have such an anti-parallel structure in the second loop. Okay, now what you see is that the hydrogen bond is just between the loops, between the oxygen of a carboxylic acid group and a hydrogen of the other side of the protein, which is close to the side which has looped. So you have such a zigzag structure which is being formed, and this is what you refer to as the beta sheets. So secondary structure, you can see it involves formation of hydrogen bonds, but you can have these two different types of structures. Of course, if you go ahead, you see some other forms being referred to, but for now, the alpha helix and the beta structure is just enough. All right, now we move on to the tertiary structure, So here too, this is just to show you the type of bonds which are involved in the formation of a tertiary structure. And that's the most important thing, because you can't be drawing all the amino acids with all the side chains. It will just make everything so clumsy. So here, okay, so Ahmed, I think you didn't hear me well, saying that you say, so what does it mean? Say the secondary structure is a primary structure that is joined in a loop. I don't know what you mean by that, but if the primary structure is only referring to the sequence of amino acids that is in the protein chain. We have seen that is very simple. One amino acid is joined to the other, joined to the other, joined to the other. You have a chain which forms a protein. You can refer to that if you count the amino acids 1, 2, 3, 4 all the way to the end. You can see which amino acid joins to which one in that order. That's a primary structure. Then for the secondary structure, you have that primary structure which you are referring to folding to have a particular form, so that if it's an alpha helix, we saw that you have a ribbon-like structure, that you will be having some hydrogen bonds between some oxygen of a carboxylic acid group and hydrogen of an amine group. Or we saw the zigzag shape, which is formed by the beta sheets. That's a secondary structure. So it's not only a loop. If you refer to a loop, the loop is what we see in the beta sheets. But if you you are looking at the alpha helix. That's not a loop. That ribbon-like structure, you can refer to it as a loop. It's some form of a twisting of the structure to give you such kind of a ribbon-like structure. Now, what you are seeing on your screen now as a tertiary structure, you can see that you are having more than one secondary structures. So it is one protein, one big protein that has folded such that you can actually see that in some portions you have alpha helix structure, and in some portions you are having some secondary structure of beta sheets. You can see that. So you see, if you look at it, these ribbon-like sites, you can refer to them as secondary structures, like the alpha helix. And you see, you have some parallel or anti-parallel side that is having some, these hydrogen bonds being depicted between them. So that's like a beta sheet. So same protein, and it's having more than one secondary structures. Now you want to pay attention to the bonds that are formed in this structure, the tertiary structure. You have the first one here being referred to as a salt bridge. So that is some form of ionic interaction between the amine group, the zwitter amine group, and you have this carboxylic acid group also in its ionic form. If you are having this amine group and the carboxylic acid groups, as how we saw them in the ionic form, in the zwitter ionic forms of the amino acids, then you can have such ionic interactions between the amine group and carboxylic acid group, what we refer to as salt bridges. Of course, you do have also hydrogen bonds in this case, where, for instance, you can have hydrogen from the hydroxyl group of, let's say, serine. Serine, if you go and look at the side chain of serine, is CH2OH. And so you see, you can have the hydrogen from a serine group and oxygen from another serine group having some hydrogen bonding interaction between them. And this hydrogen bonding is also involved in the formation of tertiary structure. And that's an interaction between two different portions, two different secondary structures. Then we also see hydrogen bonding again, which is occurring between a carboxylic acid group and hydrogen of an amine group at different points of the protein structure. This is the kind of the bond which even occurs within the alpha helix structure, what we saw earlier. You see this type of bonds, and the hydrogen bonds that exist between the parallel and the anti-parallel sides, this hydrogen bond also forming that the beta sheets. And then you have some bond. This one we haven't talked about this disulfide bonds. Where can we have disulfide bonds forming? So when you have sulfur hydral groups of cysteine. So if you go to look at the structure of the amino acid cysteine, you see that the side chain of cysteine is CH2SH. And so you can have between the two sulfur hydral groups, you can have the formation of what is being referred to as disulfide bonds, when each of those sulfur hydride groups give out hydrogen ions. Now, when you listen to this, what I'm saying, in order for you to have a better understanding of it, you have to go back and take the structures of the amino acids. And then you'll be able to understand clearly what you're referring to. If you say a disulfide bond, and we're saying that the disulfide bond can form between two cysteine molecules. So these are two cysteine residues that are found in the protein. And when the protein folds in such a way that you have this coming together, then you can have these disulfide bonds forming between the side chains of these amino acids, which are in close proximity. All right, so I see lots of these messages coming. Okay, I think it is Foley's, that is still struggling with the sound. She can't hear. Okay, she put it on speaker. Then you increase the volume and see if it will be clear. But I see she has even changed it to Bluetooth, as if you have some Bluetooth speaker or something like that, which okay, that is Charlotte. I think as for that will not work at all. I think you should rather have it on speaker, increase the volume so that you can hear. All right. Now, other bonds that can be present is hydrophobic interactions. Hydrophobic interactions. Whatever it is, the lecture is being recorded. So after this, I will share the recording with you. So those of you who are having issues with sound over the period, this is well recorded, and so you can later on watch the video and hear everything more clearly. Kelvin says, in any case, are we going to draw a tertiary structure? But why not? This is simple. I can ask you to illustrate tertiary structure of proteins, showing the different bonds that can exist in tertiary structure. I mean, the focus is to understand that you have different bonds that are involved in the formation of a tertiary structure of proteins, so that you should be able to show a salt bridge. What's a salt bridge? So you show it that it's an interaction between the ionic amine group and the carboxylic acid group of amino acids. Hydrogen bond should be able to show it. The disulfide bonds, hydrophobic interactions. So these are hydrophobic side chains groups that can have some interaction between them, like this. For instance, you have phenylalanine, these benzene rings that have this hydrophobic interaction between them. You have, say, CH3, let's take, for instance, this is alanine, and between these two methyl groups, you can have such hydrophobic interactions. The bonds which you see up here, or the hydrogen bond, this is just to show, let's say this protein is in an aqueous environment. And you have these side chains, like this tyrosine side chain, the benzene ring with a hydroxyl group on it. And you have a serine side chain, CH2OH. These can form hydrogen bonds with, say, water molecules around it. These are not. So up here is not involved in formation of a tertiary structure. It's only depicting interaction of the protein with its aqueous environment. That's what is being depicted here. But the bonds that are involved in the formation of a tertiary structure are salt bridge, that's one, hydrogen bond, that is two, disulfide bridges, that is three, and four, hydrophobic interactions. you have these 4 bonds which are involved in the formation of the tertiary structure, and which bonds are involved in the formation of a secondary structure. The hydrogen bond. I mean, so far, when we looked at the formation of the secondary structure in the alpha helix, is hydrogen bond. In beta sheets, it's still hydrogen bond. But when you come to the tertiary structure, you are seeing that you have other bonds that are responsible for the formation of the tertiary structure. And we've counted 4 of them: salt bridges, hydrogen bond, disulfide bridges, and hydrophobic interactions. So it should, Kelvin, it shouldn't be a difficult thing for you to illustrate this. The disulfide bridge, it's not a difficult thing to show. Hydrogen bonding between these groups, let's say serine CH2OH groups, salt bridge, hydrophobic interactions. These are not difficult things. All right, so Charlotte, okay. Yes, after the lecture, just like the last time, you have all the materials. So if there has been any problem with sound, you couldn't hear and all of that, you can listen to the recording. And you should also know, like on my YouTube channel, there are some videos there already before I add these new ones. So if you are really engaged with the material, you study hard, further, and all of that, you have all the opportunity, because there are things I may say in this video which I haven't said in the older videos, and vice versa, so that, I mean, you have the opportunity to watch same lecture, which maybe last year, or if I don't know if last 2 years there's anything there, but maybe last year, and you listen to that one and listen to this one, and you combine. So you have further information. You have the presentations. I sent you a whole book the last time. So if you use the presentation slides to guide you, you can read specific portions of that book and listen to the video, make notes, and you shouldn't have any problem at all. All right. Then a quaternary structure. So here we are bringing all of these together. We are bringing all of these together, where you have a primary structure, which is just a sequence of amino acids in the structure of the protein. And from there, you have the formation of a secondary structure, which can either be an alpha helix or a beta sheet. Here, the secondary structure that is being shown is an alpha helix. And from that secondary structure, you can have a protein that is having more than one secondary structure in it. So like this is a tertiary structure, which is having a protein having some alpha helix at some point, and you have also some beta sheets that can also be present there, where you can have the folding of the protein to have a particular form in space. So in such a conformation that makes that protein to be active and carry out its function. And if you have proteins with more than one tertiary structure, so you can see here, this is a hemoglobin molecule that is having 4 different subunits, together, having interaction between them, acting as one unit, carrying out a function. That's a quaternary structure of protein. Right. Now we can refer to what we refer to as denaturation of proteins. What is that? What is denaturation of proteins? Now, don't confuse denaturation of proteins to think that it is the breakdown of proteins into amino acids. No, that's not what it is. Denaturation of proteins does not result into a breakdown of proteins into amino acids. I mean, a breakdown of proteins into amino acids, that's metabolism, which is catabolism, breaking down of the protein into amino acids, and those amino acids will then go into the synthesis of proteins which are required in a particular organism. But that's not what denaturation is about. All right, let me see. There's Mujiba says, sir, please, not all of us had a slice. For example, I tried to link the ICT on campus through email to help me resolve my team password. They haven't gotten back to me, so I'm trying to go back. So if you have any problem, you shouldn't even be able to be here in the first week. So you don't really have any problem with ICT, because the link is easy. You just tap on it and you join the class, so there's not a problem. And if he's talking about slides, it wasn't shared through here. I gave it to the class rep, and the class rep should have shared it in your group. So maybe you are not following what is happening in the group. That's why you didn't have it. Okay, so all right, somebody say I should repeat what I was saying about denaturation. Now, you see, we talked about proteins having secondary structure, tertiary structure, quaternary structure, so that a protein which is having a particular conformation as a tertiary structure or a quaternary structure, it is in that form that the protein will be active to carry out its function. Now, you see, we refer to these hydrogen bonds, disulfide bonds, salt bridges, hydrophobic interactions. Now, those bonds can easily be broken down by elevated temperatures, heat. They can be broken down with decrease in pH. That is in a harsh acidic medium. Those bonds can be broken down, or in harsh basic medium. You can have those bonds being broken down, because each of these proteins have optimum conditions within which they stay in their conformational structures. You can have even agitation. For instance, what is being depicted here, like egg yolk. The egg yolk is having such a form, semi-solid, because of those bonds that exist between the protein subunits. That is why you having such a particular form of it. But if you beat the egg, if you beat the yolk, it scatters. It becomes more fluid. You would have broken down those hydrogen bonds and disulfide bridges. All those bonds would have broken, and that yolk would have lost its form as it was before. And that is denaturation of the protein. The protein will no longer be able to carry out its function because its functional conformation has been broken down. Like for an enzyme or some hormone or a neurotransmitter that is having a particular form, if those bonds that are involved in the formation of those secondary, tertiary, and quaternary structures are broken down, you have a denaturation of that protein. That's what is being referred to as denaturation, and not when you have proteins broken down into amino acids. That's metabolism, catabolism, breaking down of proteins, which is different from denaturation. Paul Abochi says, did you say that the quaternary form that we see is, I don't know what you mean, in its quaternary form that we see its functions? Okay, what I'm saying is that proteins will have, so a particular protein is either having usually it's a functional proteins. Let's say if you say an enzyme, that's a protein, or you say a hormone, that's a protein, or a neurotransmitter, that's a protein. These are usually big proteins that are folded in some particular conformation, and it is in that conformation that they carry out their function. So that if that conformational structure is broken down, then the protein will be non-functional. So that's what I'm referring to. So that you need a protein to be folded in a particular form in order for it to be active, because you have particular parts of the molecule that will have to be in close proximity to be interacting in order for that protein to carry out its function. Right. Now, proteins, as we speak of these proteins, you can have proteins carrying out different functions. And the common functions of proteins include proteins acting as enzymes, and these are biological catalysts. These are molecules that increase the rate of biochemical reactions, in biological systems. We'll take a closer look at this function of proteins as enzymes. You have some proteins which are, structural proteins, forming body tissues such as muscle, the hair and the nails, say of keratin. These are structural proteins. You have some proteins which are embedded in cell membranes, which are responsible for the transportation of substances into and out of cells. You even have some proteins which are embedded in cell membranes that act as receptors on cell membranes. These receptors are for for cellular response or for cellular communication. I think that we'll take a look at it in the second year. Looking at cellular communication, we'll come across proteins acting as receptors. Mostly, they are going to be in complex with, say, carbohydrates. So you have like glycoproteins, or you have in complex with lipids, like lipoproteins. And these will be acting as molecules on the surface of cells. Some of them will even be in the cytoplasm, responsible for communication between cells. You have, proteins, acting, so particular hemoglobin, which is acting in a particular function of transport of oxygen and carbon dioxide, that is gaseous exchange in the circulatory system, in the blood circulation system of the body. These two, we will look at it much further. Within the context of this course, we will look at, the first, which is enzymes, proteins acting as biological catalysts, much more into detail, and then hemoglobin, where you have the function of protein for oxygen transport. We'll look at these two more closely. Others, like, proteins acting as transporters across membranes and receptors in cells, I think that will be in second year or so. And you also have proteins that can be acting as neurotransmitters for signal, transduction in the nervous system, or what you refer to as white blood cells. If you look at it much more closely, you come to see that they are proteins. Those are proteins. And so, proteins, which are being referred to as immunoglobulins. And you have some of these proteins that are for regulation of biochemical reactions in biological systems, also being referred to as hormones. So, biochemistry is that interesting that if we take only proteins, for instance, this can give you different branches of specific fields. For instance, if it is the first one, that can take you into a whole different specific field, what is being referred to as enzymology, which people specialize in. You can specialize in enzymology, do masters, do PhD, particularly a PhD in enzymology, and become a professor in enzymology, for instance. Or you have if maybe if you look at say hormones, for instance, that will go into a field of say endocrinology, for instance. Or if you have proteins, forming immunoglobulins, which is particularly for this immunologic defense. That is a whole different field again that is being formed there. Immunology can have that. And you have this in cellular biology in general, you will see you will come across these studies of proteins as transporters in cell membranes, and also as receptors in cell membranes, like in cellular communication. You come across that. For structural biology, where you're looking at these specific proteins that form body tissues, like muscles, structures in the hair, the nails, you see such. So you can see how really broad biochemistry is, such that it can take you into some very specific fields in the future. Irene says, is hemoglobin or hemoglobin? All right, the first spelling is American, and the second spelling is British. So it's the same. If you have if you take a dictionary, it can give you both of these spellings. The first one, you will refer to it as an American spelling, and the second spelling is a British English spelling. All right. So if you write any of these, it's correct, except that it's good practice that when you are writing, if it is an American English you are writing, you write everything American English. And if it is British English you are writing, you write everything with British spelling. You don't mix them. That is good practice. Okay, now what we do is, I think today we can start to look at this function of proteins, the catalytic action of proteins, proteins as enzymes. We can quickly get ourselves introduced to this particular part, and then the next time we meet, we'll continue with it. And then when we finish with that, then we take on the structure and function of hemoglobin. And then we'll be done with that function of proteins there. All right. So we continue with the enzymes. And what are enzymes? What are enzymes? So I was just saying that enzymes are biological catalysts. They speed up, biochemical reactions. And why is it needed? Why do we need enzymes? So let's take a simple process of metabolism of carbohydrates, say sugar, glucose. We usually do talk about metabolism of glucose, and we say glucose is for energy generation, for instance. No, we are done, Ahmed. We are done with the amino acids. We are done with them, the 20 amino acids. So we continue then. We have even formed proteins out of it. Out of that, we've talked about formation of peptide bonds to form proteins. And even the proteins, we've looked at it at different levels: primary structure, secondary structure, tertiary, and quaternary. So we are done. All right. Now we see there's a simple reaction on the slide on the right-hand side that this reaction represents metabolism of sugar. I mean the tabletop sugar. That is sucrose. Sucrose. And sucrose is a disaccharide of glucose and fructose. Now, if you take in this sugar, the simple reaction here, this is very much of a simplification. Simplification of the breakdown of sugar to give energy. And so you have sucrose undergoing some oxidation as oxygen, like a cellular oxidation, such that there's what we say carbon dioxide is a byproduct which is formed from cellular respiration. Some water is also formed, and then energy is generated. Now, we talk of these as sugar giving you energy, source of energy, say carbohydrates giving you energy. But if you think of it, if you have sugar, I mean sugar in a container, and you place it in your kitchen or on your dining table, that sugar is there for a very long time. But the sugar is exposed to oxygen, all right? After all, is there not oxygen in the environment? So how come the sugar does not change into carbon dioxide and water and energy is generated? If you leave it there, the next day you come and see it. A week, the sugar is still there. A month, the sugar is still there. The sugar does not change into carbon dioxide, maybe some water vapor, and then energy generated, and the sugar vanishes and you don't see it again. That's not what happens. But the moment you take this sugar into the body, within milliseconds, that sugar is broken down and generating energy, and you have formation of carbon dioxide and water very, very fast. That reaction occurs. How does that happen? That happens because of the presence of enzymes. Enzymes accelerate, they increase the rate of the reaction. That is why you have such a thing happening. And why should the reaction be sped up? So if you also think of it logically, in the body you have a lot of biochemical reactions that are happening. So for instance, as I speak to you, my jaws are moving, my brain is coordinating all the activities as I speak. My eyes can see on the slide. I see the thing and then I speak. My mouth, the muscles, everything is being coordinated. If I make any gesture, even with my hand, your ears, as well as my ears, because as I speak, I hear myself as well. You have sound that is, hitting your eardrum, and that sound is being transmitted to your brain, and you have understanding of what is being said to you. All these things are happening at the same time. All of this, your heart is beating, your kidneys are working, your liver the organs all working at the same time. So you can imagine the numerous amounts of chemical reactions that are occurring in your body at the same time. And all these reactions need to be coordinated. So you can think of it as your body being a very complex chemical factory in which there are various complex chemical reactions, complex and simple chemical reactions that are occurring. And all of these reactions have to be coordinated so that if there is any problem anywhere, so for instance, maybe the coordination between my speech and my hearing is, that is in the brain, begins to falter, then maybe when I speak, I can't hear myself well, and so I cannot time my speaking. And you will see that there will be some kind of a discoordination, a discordance between my speech, and even if I do gestures, maybe the control of the muscles of my hands, and so maybe I can't move my hand the way I should move it according to what I'm saying. Maybe my speech can even be either very slow or too fast. So you can look at this in this way to see how very important it is for all these chemical reactions to be running, very fast, such that they get coordinated with one another. Now, if you were running a chemical reaction in the laboratory and you wanted to increase the rate of the chemical reaction, the simplest thing you could do is to increase temperature of the reaction system. Because, that is just agitation of molecules of the reactants so that these reactants will quickly react. That's what you do in the lab. You increase temperature, you add some heat. If you go into, let's say, microbiology or the chemistry lab, you do these things where in microbiology, for instance, you do some things like incubation. You put some media in a particular temperature condition, or you in the chemistry lab, you run some reactions, you apply heat because you want it to occur very fast. You agitate the molecules, you give more energy to the molecules to react. But you cannot do that with biological systems. As you see, we just talked about denaturation, where increase in temperature can result into the breakdown of proteins, and these proteins will lose their functions. And it's not only proteins, even nucleic acids, DNA, RNA, these all can break down. And so that's not, it's not an option for biological systems to increase temperature in order for these chemical reactions to occur very fast. And so we have these enzymes that do this for us. So this is how very, very important enzymes can be in terms of, keeping chemical reactions at an optimum rate that makes life sustainable. Anyway, I don't know, Kira, if you did science in senior secondary school. If you don't understand, it only means that you have to sit some more and study. So somebody says, is it that all proteins are enzymes, but not all enzymes are proteins? Yes, I think that statement is here, that you have a small group of RNA that we refer to as ribozymes that also act as enzymes. And so, yes, your statement is right. So that is, who said that? Christopher Nin Tetelate, yes. Your statement is right. And then Kira says, honestly, when we come face to face, there will be a lot of questions from my side. No problem. you can ask all questions you want to ask. You can ask here by writing in the chat. You can raise your hand. I will give you the chance you speak, and I refer to you. So it's not a problem. Okay. All right. So I think we can continue. So I mean, it's a lack of enzymes and all of these. These are things which I think when we I don't know if in your course, I don't know if we'll come across some, we'll look at some of these deficiencies and how some of these, there can actually be some synthetic analogs of some of these enzymes that are taken in to correct some of those the issues you are thinking about. I mean, it's just interesting that quickly you start to think in that line. That's what actually university studies of this nature should do to you. You should think further into some of these things. And I'm happy that you can quickly think about something like that. So lack of enzymes, for instance, how that will result into some medical condition. How will you deal with it? And then you begin to look for alternatives. So yes, those things happen. You have some lack of or deficiencies. And if you have deficiencies, you can get some analogs of these enzymes. So that is in the pharmaceutical industry, to solve some of these problems. And Bismarck says you feel it like you want to die, but you will not die. Is that clear? Oh no, I don't think that's what it is. That's not what it is. Maybe just some particular. There are numerous enzymes in the body, so that a deficiency of one will result into some metabolic disorder, and such a metabolic, like say maybe how do you call it? Lactose intolerance, for instance, which is a deficiency of lactase, the enzyme that breaks down milk sugar lactose, for instance, which is a lack in such persons, which then will cause that lactose to remain as a undigested part of food, which when it gets to the large intestines, then you have intestinal microbiome acting on the lactose and resulting into bloating due to the formation of more and more carbon dioxide. You have gas, which will be making you feel bloated, discomfort in the digestive system. But like the question he has asked, what you can do with that is say in food production itself, you have the introduction of say lactase into the food product to break down lactose, so that say for instance a milk, you have milk which the lactose intolerant persons can consume. That milk is infused with lactase to break down the lactose in the milk, so that it does not go to cause that kind of discomfort, that metabolic problem for such persons. So you see, that is that what your friend was actually thinking about is something very, very practical. I mean, what that's Mujib. Yes, Mujib is a very, very practical thing that he's thinking about. All right. Okay. So, Adam says, no, I always give self-assessment questions to students. So that one, I even said it from the beginning that I'll give you self-assessment questions. So don't worry about it. Over time, I'll give you those self-assessment questions. So you will surely have them. All right. Now we can continue. We can continue from here, having that introduction on enzymes. We can talk about another very basic aspect of the action of enzymes, and this is a principle for all chemical reactions. All chemical reactions. Now, for every chemical reaction to occur, that will result into the formation of products from reactants. You have this principle that, let's take for instance where you are now, where you are sitting, where you are lying down, whatever position in which you are, and listening, watching. If you are comfortable in a particular position, there will not be any need for you to change to another form, to change to another position. You will continue to be in that position. It is only when you feel a little discomfort that you want to change the position in which you are sitting. You always want to find the most comfortable position to be in, the most stable position to be in. And this is so even at the molecular level and even at the atomic level. So that reactants, these are molecules. What you see on the left-hand side as activation energy profile, you have on the vertical axis energy, and you have on the horizontal axis a reaction coordinates. Now, what this means is that at a particular energy level, you have reactants. These reactants separately are in stable forms. I mean, molecules are in their stable forms at a particular energy level. Now, these molecules, these reactants, in order for them to, react to form products, they require what is referred to as activation energy, which is to, take these molecules to an energy level that they are unstable, so that at that energy level, where they are unstable, what we refer to as the transition state, they begin to look for a more stable energy level. So that's when they react to form a compound, and that compound is the product which is even at a lower energy level. So you see that on the vertical axis, you have products at a lower energy level, where you have these products in a much more stable state. What you see with this energy profile is that we have two curves. One curve is blue, and this curve refers to an uncatalyzed reaction. And you have the second curve, which is brown or orange. This is the curve for a catalyzed reaction. And because we're speaking of enzymes, particularly, we can say it's an enzymatic reaction, to say that enzymes are catalyzing this reaction. And what you notice is that the activation energy for the catalyzed reaction is lower than that of the uncatalyzed reaction. So this is the mechanism by which enzymes increase or accelerate the rate of chemical reactions. so that without enzymes, you have a large amount of energy which is required to activate reactant molecules to reach a transition state before they react, and then they form the product at a lower stable energy state. But you notice that in the enzymatic reaction, you have the enzyme which binds to the substrates to form an enzyme substrate complex. What you see as ES complex, that is an enzyme substrate complex, which is a slightly lower energy level than that of the reactants. And then the energy which is required to move that enzyme substrate complex to its transition state is much, much lower than what you see in the uncatalyzed reaction. So what does it tell you? It tells you that because you have the activation energy for enzymatic reactions being much lower, the reaction can proceed to the formation of products much faster than for the non-enzymatic reaction. Now, with this principle of activation energy profile of chemical reactions, comparing that of an enzymatic reaction and that of a non-enzymatic reaction, you can understand why enzymes are able to increase the rate of chemical reactions. All right. So of course, this you can also even think of it this way. Okay, so it means that is why you can have the reaction running at a much faster rate, even though you don't increase temperature. So you see, the reaction without the enzyme will require higher energy. Like what I was saying, say these physical reactions which you run in the lab where you heat a reaction mixture for the reaction to occur fast, you see, you require much, much higher energy. But in the enzymatic reaction, once you have the enzyme substrate complex, you need lesser amount of energy to have this complex go to a transition state and come into form a product. Right. Now, before we continue even looking much more at enzymes, one of the things we should do is to understand some basic terminologies that are used in enzymology or in the study of enzymes. Because we do refer to these things when we speak, we use these terms, and so it will be good to understand what these things are even before you go further, because every subject has its language. And such vocabularies, once you understand them, then you'll be able to understand and communicate better in that particular field. Now, first is cofactor. What is a cofactor? So you see, we did refer to the fact that most enzymes are proteins. Even though you also want to remember that you have some catalytic RNAs that also have, such enzymatic activity. You have cofactors being non-protein parts of enzymes. These are required for that protein part, the enzyme, to be active. Usually, when we say cofactors, this refers to metal ions. We'll go further and look at some of these cofactors, and you see these are metal ions that these proteins require in order to have their conformational structures that are active. In some biochemistry books, you may see that the term cofactor is used interchangeably with coenzyme, but in some you will see that the distinction is drawn to make it much clearer to you that Coenzymes are also non-protein, but these are organic molecules. So that you see the cofactors, I said these are metal ions, like you say ferrous ion, like Fe2 plus, or say nickel, copper, magnesium, all those ions, these metal ions, they act as cofactors. But when we say coenzymes, these are organic molecules, and they are usually vitamins. So the vitamins we take in, particularly vitamin Bs, these are precursors. So we can say that those vitamins that we take in, like you say you are taking in vitamin C, the form in which you are taking it is an inactive form. But once it gets into the body, it gets converted into its active form, usually by processes of, say, phosphorylation, for instance. And you have, or say, binding with some derivatives of nucleotides, some coenzyme A, and then you have these, or even some binding with some derivatives of some amino acids, and you have the active forms of these vitamins, and they bind to these protein parts, which then will give you a full enzyme. These are what we refer to as coenzymes. They are not metal ions, they are organic molecules, but they are non-protein that are in complex with a protein part acting together as an active enzyme. Then you have apoenzyme. So the apoenzyme, which is even sometimes just referred to as an apoprotein, which is to say that protein portion, the protein portion to which the non-protein portion is bound before you have an active enzyme. That's an apoenzyme, or you can call it an apoprotein. All right, so Mujiba again says, where will you classify vitamin K? Is it a coenzyme or a cofactor? Oh, we don't do such a vitamin cannot be a cofactor. So let's get that one clear. As I just said, cofactor will be metal ion. I mean, usually these are metal ions. And coenzyme is an organic molecule. So if a vitamin is going to be a coenzyme at all, if a vitamin is going to be acting as an auxiliary part of an enzyme, it will be a coenzyme. A vitamin will be a coenzyme and not a cofactor. Okay. Now, vitamin K. Yes, vitamin K also acts in some very, different reactions in the body, acting, I think, for instance, in blood clotting, for instance, you come across vitamin K carrying out, some functions, some participating in a cascade of reactions, different reactions, one after the other, which result into blood clotting, for instance. In such an instance, it is not a cofactor. Like I said, a vitamin is not a cofactor. If anything, if it is acting as an auxiliary of an enzyme, it is a coenzyme, not a cofactor. A cofactor specifically refers to metal ions. So when we come to that, you see, we actually will go further and look at examples of these cofactors and coenzymes. So you see them clearly. But even in this case, vitamin K will be carrying out some activation processes, yes, of some enzymes. You may not refer to it as a coenzyme because it is actually not bound to an enzyme as part of an enzyme. All right. Inhibition. Okay, no, I think the next thing is an active site. So I think that is even depicted on the left-hand side in this diagram, where you have, say, what is being referred to as an apoenzyme. There is a certain group which is shown in it. You know, if you take a protein, that protein, we have just talked about the structure of a protein having a primary structure, secondary, tertiary, quaternary. That tells you a protein can be a big molecule. Various amino acids coming together to give you a protein. It's a large molecule. And if it should be interacting with some compound, it is not just on the whole surface of the protein that you can have these compounds interacting, but there's some specific portion of this protein molecule that will be having interaction with a particular molecule. Say the substrate, what we refer to as a substrate, is the compound that is involved in the chemical reaction upon which the enzyme is acting. That's a substrate. So, for instance, if you take an enzyme, say, you say salivary amylase, that's an enzyme, salivary amylase. And you say the salivary amylase acts on starch. So, starch is the substrate. It is on starch that the reaction, the breakdown reaction is occurring. So, that becomes a substrate. That last term there, the substrate. Now, if you take the salivary amylase as a molecule, it is not on its surface, the whole surface anywhere at all that the starch molecule can be having an interaction. But some specific portion, some specific, maybe 2 or 3 of amino acids that when the protein folds, they come together, form something like a pocket into which the starch molecule gets into for the reaction to occur on it. And that portion of the enzyme that is having interaction with its substrate is what is referred to as the active site. The active site of the enzyme. Now, any process that results into an increase in the activity of an enzyme is an activation process. And any molecule that does that is an activator. So, for instance, a cofactor or a coenzyme is an activator. It activates an enzyme. And you can have molecules that do the opposite. They will inhibit. They will make an enzyme inactive, either completely or reduce its activity. That is an inhibition process. That's what is being referred to as an inhibition. And any compound that does that is an inhibitor. And you also, as you go further, you come to see that it is this knowledge which is used in pharmacy, such that if a compound is known to inhibit the activity of a particular enzyme, or so if a particular chemical reaction is to be controlled, then you can use the inhibitor. So that when the inhibitor is taken in, the inhibitor goes to inactivate the enzyme. This can be used against, say, bacteria, for instance, in infections. All right, so these terms, once you understand them when we speak, you will understand the language of enzymology. Cofactor, coenzyme, apoenzyme, active site, activation, which is caused by an activator, inhibition, which is caused by an inhibitor, and a substrate. All these terms should be clear to you by now. And when we do refer to them, you will understand. Now, another simple fundamental thing that we should also know is how enzymes are named. How enzymes are named. So there are some simple ways in which enzymes are named. The first one is adding the suffix ase, A-S-E, to the name of the substrate on which the enzyme is acting. So, for example, you have the enzyme urease, and you notice the suffix ase, this A-S-E, ase, is added to the name of the substrate, which is urea. So urease catalyzes the breakdown of urea. And so we only add ase, A-S-E, to the name of the substrate on which the enzyme acts. The same thing you see with the enzyme lactase. So lactase is an enzyme which catalyzes the breakdown of lactose. Lactose is milk sugar. And so that is the substrate. Lactose is a substrate. So we only change this end, the lactose, we change the end, at the suffix ase in place of ose, and that gives you lactase as the enzyme. So Irene, exactly what you have written, exactly as it is. And I'm happy you just quickly picked that and you understand it. Very good. Now, another way that enzymes are named is by the activity or the reaction that they are catalyzing. Remember, what we just looked at before is based on the substrate on which the enzyme is acting. This time around, it's not on the substrate, but on the reaction, on the activity. So, for instance, you hear a name like DNA polymerase. What does DNA polymerase do? It catalyzes polymerization of nucleotides. That is to say that it catalyzes the bonding of nucleotides. So nucleotides are the basic units of nucleic acids, say DNA and RNA. Just the same way as we have just talked about amino acids that bind together to form proteins, you have nucleotides also binding to form nucleic acids like DNA and RNA. And in this particular case, the compound which is being formed, the polymer which is being formed, is DNA. And it is being formed by bringing together nucleotides. That is to say, polymerization of nucleotides. And so the name is polymerase. That's the reaction, the activity, polymerization. Same way, you have an enzyme, pyruvate decarboxylase. Now, the substrate is pyruvate, and the enzyme removes carboxyl group. So that's why I say decarboxylase is removing carboxyl group, COOH. You know, the carboxylic acid group which we saw in amino acids, similar group that is being removed from pyruvate. And so the enzyme's name is pyruvate decarboxylase. You also will see a name like, say, lysozyme. And that is involved in the breakdown of bacterial walls. This is one of these principles which is used in the production of some antimicrobial agents to break down bacterial walls, which then will result into bacterial death. That breakdown of the bacterial walls is a lytic reaction, what is referred to as lysis. It's a process of lysis. And because of that activity, lysis, the enzyme, so this time around, is not the addition of the suffix ase, but zyme from enzyme, which is being added to have what? Lysozyme. So Mujiba is saying, so under what conditions can an enzyme be destroyed? Don't worry, don't run before the gun. We will come to conditions, optimal conditions of enzymes. So when we come to optimal conditions of enzymes, then you will see specific factors that are required for the integrity of an enzyme, so that if those factors are disrupted, it can result into destruction of an enzyme. So don't worry, we'll come to it and you'll see it. Now, apart from these two systems of naming enzymes that we just saw, that is the first, naming by substrate, the second, naming by the activity, you also do have some enzymes that have common names. These do not go according to these two systems that we just referred to. So for instance, for some of these first enzymes which were discovered, like pepsin, the name pepsin comes from the Greek word pepsis, which means digestion. And that is because pepsin was discovered and known to be acting in the process of digestion of foods, because it was first extracted from the digestive system, from the digestive juice. And so then it was known that it was involved in digestion, which is why it's being referred to as pepsin. Another one, trypsin, was discovered by rubbing pancreatic tissue with glycerol. We just took glycerol and use it to rub pancreatic tissue, and you have the extracts, the glycerol extract containing this enzyme, trypsin. And because of that process of wearing down the tissues of the pancreas, that resulted into the extraction of this enzyme. The name is given trypsin, coming from the process in Greek, trying, which means to wear down. So these are common names which for these first enzymes which were discovered. You don't have such a system that we just referred to for naming enzymes. Right. So that is about naming of enzymes. We continue to look at classification of enzymes. Now, enzymes can be classified, and I think we'll end with classification of enzymes today. Enzymes can be classified into 6 main groups. The first is what we refer to as oxidoreductases. And as the name suggests, these are enzymes that catalyze oxidation and reduction reactions. The usual, types of reactions which we refer to as redox reactions, oxidation reduction. There are a lot of biochemical reactions which are these redox reactions. And the enzymes that catalyze those reactions are what are being classified as oxidoreductases. And you have an example here where you have the conversion of lactates or lactic acid to pyruvate. And you can see that there's the carbon, the central carbon there is having a hydroxyl group on it. That you have that oxidation on that carbon, so that hydroxyl group is converted to a ketone group. So you have a C double bond O there. That's oxidation. If you take the reaction backwards, that will become a reduction. You always have oxidation and reduction occurring at the same time. So you have that enzyme, lactate dehydrogenase, as an oxidoreductase. So the enzyme lactate dehydrogenase is an oxidoreductase. All right. And in fact, as you go ahead, you will come to realize that this term, dehydrogenase, sometimes is even used interchangeably with the word oxidoreductase. So that in classification of enzymes, sometimes this first group, which is oxidoreductase, is also referred to as dehydrogenases. You will see that also in some biochemistry books. Right. So that is it. First group, or first class, oxidoreductases. Second class, transferases. And these are involved in the transfer of functional groups from one molecule to the other. This is also a common type of reaction you will see in biochemical reactions, where you have one group of one functional group from a molecule that is being transferred onto another molecule. And here we see an enzyme, aspartate aminotransferase, which sometimes is also referred to as aspartate transaminase. And this is involved in the transfer of functional groups. So we see aspartate or aspartic acid, that's an amino acid. And we see alpha ketoglutarate, that is a carbohydrate. I mean, if you go in, I don't know if you did metabolism of carbohydrates, talking of what you refer to as the Krebs cycle or the TCA cycle. You will see these intermediates in that cycle, in the breakdown of carbohydrates to generate energy. You see alpha ketoglutarate there. Now, your attention should be drawn to the shaded portions where you have an amine group in aspartic acid, the NH3 plus there, shaded yellow, and you have this ketone group, the C double bond O, shaded red in alpha ketoglutarate. Now, what you notice is that there is an interchange. So these two are reacting, and what is really the reaction? The reaction is a transfer of functional groups, so that you see that this amine group is transferred to the position where you have the C double bond O group, the ketone group, and vice versa. The C double bond O, that ketone group, red, is also transferred to the position where the amine group is. And so the reactants become oxaloacetate, where now you have this position where the amine group was, you now have the C double bond O there, and a position where you have the C double bond O, you rather have the amine group coming there, so that you now have glutamate. So aspartate is converted to oxaloacetate when the amine group is interchanged with this ketone group. And alpha ketoglutarate is converted to glutamate or glutamic acid when you have the ketone group, the C double bond O, interchanged with the amine group. So you can see that transfer of functional groups. And the enzymes that are involved, the enzymes that catalyze this reaction, which is the aspartate aminotransferase or aspartate transaminase, is classified as a transferase. All right, so Charlotte says what? Enzymes work under optimum temperature, high temperature destroys enzyme, vice versa. Correct. All right. Now we have the next group, the next class being hydrolases. Hydrolases. These are enzymes that catalyze hydrolytic reactions. So you have the breakdown of molecules with participation of water, being referred to as hydrolysis. And the enzymes that catalyze these reactions are hydrolases. And an example here is acetylcholine esterase. And what does acetylcholine esterase do? Acetylcholine esterase breaks down acetylcholine into acetic acid and choline with participation of water. And so you can see that this is acetylcholine, the big molecule there, broken down into two molecules. So the acetic acid side, and then you have the choline side. That enzyme, acetylcholine esterase, that catalyzes the reaction, is a hydrolase. Next, the fourth class, lyases. Lyases. And what do lyases do? Lyases either add or remove groups from a molecule. If the lyase is involved in the removal of a group, a functional group, from a molecule, it results into the formation of a double bond. Or if it does the opposite, where there's a double bond in a molecule and the enzyme rather catalyzes a reaction of addition of groups, the double bond is removed. So that double bond becomes a single bond. And so you see, this is another reaction which you also find in the TCA cycle, that is in carbohydrate metabolism. You realize in this compound, cis-aconitate, there is a double bond between these two carbons. Now, this double bond is removed when you have addition of water. So you don't confuse this with hydrolysis. Hydrolysis, when you see participation of water, hydrolysis results into the breakdown of one molecule into two molecules. In this case, you see that the water molecule rather breaks down into a hydrogen ion and a hydroxyl group, and that hydrogen ion is added onto this carbon. So you have a CHCOO minus on that carbon, and you have the hydroxyl group from the water coming onto this carbon, so that hydroxyl group is here. So you have a CHOH on that carbon. And you notice that the double bond, which is between these two carbons, is then converted into a single bond. So the enzyme aconitase, which catalyzes this reaction, is a lyase. And this reaction could occur in the opposite direction as well, whereby water is removed. Let's say that is a dehydration reaction, where you have isocitrate forming cis-aconitate. So that will be single bond now being converted to a double bond. The enzyme involved is a lyase. Now you have the fifth class being isomerases. Isomerases. Now, isomerases are involved in the formation of isomers. So, for instance, if you have the formation of fructose 6-phosphate from glucose 6-phosphate, the chemical formula for these two structures, glucose 6-phosphate and fructose 6-phosphate, the same, but their chemical structures are different. So you see that glucose 6-phosphate is having a 6-sided ring, and you have a fructose 6-phosphate having a 5-sided ring. But if you count the number of carbons, count the number of oxygens, number of hydrogens, phosphate group, they are the same. They are equal. It is the structures that are different. And so the enzyme phosphohexose isomerase, even in the name, the term is there, isomerase. Isomerase that's an enzyme that is involved in the formation of isomeric forms of compounds. And then the last one is a ligase. And sometimes you do see ligases also being referred to as synthetases. And these are enzymes which are involved in the joining of molecules to form larger molecules like DNA ligase, which is involved in the synthesis of nucleic acids. Or even if you have the synthesis of proteins, where you have amino acids joining together, you have the enzymes which are acting in these processes referred to as ligases. So these are the 6 classes of enzymes that we have, which also you can easily remember just by, say, acronym, OTHLIL, say OTHLIL. The O is for oxidoreductases. The T is for transferases. The H is for hydrolysis. L is for lyases. I is for isomerases. And the last L is for ligases. So that way you can easily remember these 6 classes of enzymes. All right. Now, so it says, so Mujiba said, what's the meaning of synthesis? Synthesis actually is the same as what you do refer to as anabolism, which is building up. It is the opposite of catabolism, opposite of breakdown is synthesis. You bring molecules together to form a bigger molecule. That is synthesis. All right. Now, we can also speak of quickly about 4 major classification of enzymes depending on specificity of these enzymes. The first is what we refer to as absolutely specific enzymes. These enzymes act on only one particular substrate, only one. So some of these enzymes that the example we saw, say lactase. So imagine that lactase was acting on some other substrate and you give it the name lactase. Of course, that's going to be misleading. It is because lactase acts only on lactose. That is why we can name it lactase. So it is absolutely specific to lactose. Similarly, you have glucokinase, also specific to glucose, no other molecule. And here to quickly, I want to say that if you see in the name of any enzyme, kinase, keep in mind that a kinase is an enzyme which is involved or which is catalyzing the transfer of phosphate group from one molecule to the other. So for instance, if you have transfer of phosphate group from ATP, adenosine triphosphate, to glucose, to have glucose 6-phosphate. The enzyme is a kinase. So a kinase is an enzyme that catalyzes transfer of phosphate group from one molecule to the other, which means it's a transferase. Kinases are transferases. The next, group-specific enzymes. Group-specific enzymes. Now, these enzymes will act on similar molecules with the same functional group. So, for instance, if you say hexokinase, you see what we just saw glucokinase, and we said it was absolutely specific because it will act on only glucose. But here we say hexokinase, which means it will act on hexoses. Hexoses are 6 carbon sugars. So that's a group, not a single molecule. Same way, pepsin will be acting on peptide bonds in which you have participation of amino acids with aromatic side chains. And what are those amino acids with aromatic side chains? We saw the phenylalanine, we saw tyrosine, and we saw tryptophan. So then you see pepsin acting in the breakdown of the peptide bonds with these amino acids. Trypsin is also involved in the breakdown of peptide bonds that are between carboxyl group of the amino acid lysine and arginine with another amino acid. So that's group specific. Third, you have bond specific enzymes. Bond specific. So you have, say, alpha amylase. So we human beings, we have alpha amylase. Our salivary amylase is alpha amylase, which is involved in the breakdown of the bonds between the glucose molecules in starch. And the blood bond is a glycosidic bond, which is an alpha 1,4 glycosidic bond. But the same salivary amylase cannot break down beta 1,4 glycosidic bonds, which you find between same glucose molecules, but in, say, cellulose, which is the undigestible part of food. And so we refer to that as roughage. So it's a specific bond on which the enzyme is acting. That's bond specific. And then the last one, stereochemically specific enzymes, which will be acting on, so we did see some stereoisomerism the last time, where you have D and L isomers. So you have, so for instance, L amino acid oxidase, which will be acting on only L amino acids. Similarly, you have D amino acid oxidases, which will be acting on only D amino acids. So these are specific to some stereoisomer of a particular substrate. So this is where we end our lecture for today. We have been very interactive. That is good. You keep writing in the chat. I keep responding to it. And so as it's time, I can only say thank you for your audience, and I wish you all the best till we meet the next time. Bye for now. Ahmed says maybe this is a master's course. No, it is not. This is fundamentals of biochemistry. So it's not a master's course. The only thing is that you did biology and you did chemistry, and you never did biochemistry anywhere. It is the first time ever you will do biochemistry. You do biochemistry only when you come to the university. So the things are all new. So it's understandable that sometimes you may find difficulty in breaking out of the senior high school, knowledge that you have. You have to push yourself. You have to push yourself because you did biology, you did chemistry, but it's a combination of biology and chemistry. And so you have some of these, difficulty, which is well understood. So just keep your head up and work hard, break yourself off a little bit. You have to, you know how when you are moving from one class to the other, it is always, some kind of a struggle because some new things are added. So you have to really sit up. All right. All the best. So we meet next time. Abochi, as always, I will send you the presentation slide. So don't worry about that.